What I'm going to do now is to take you through a case very briefly, and then we'll try a couple of other engagement exercises. But this is one that fits into one of the concepts called dual use. It's about possible misuse of science, and I put the in quotes, possible in quotes. And it's about the H5N1 virus or avian flu. And it's about some research that has become controversial and involved a lot of discussion with regulators. And the avian flu virus is a perfect example for discussing responsible conduct of science. So I'm going to talk very briefly about some of the details of the experiments, but not in too much detail. Hopefully at the end you will understand a little bit about the experiments, why this might be considered as dual use. And by dual I mean one side or the other, appropriate use or misuse. And this is what dual use means. And then have a look at the role of scientists in contemporary society. The work on avian flu has generated much commentary and exercised regulatory agencies in many countries. The H5N1 virus, or bird flu, is highly pathogenic. It's mainly found in birds, and it's particularly dangerous and lethal in domestic fowl. There have been a few sporadic human infections with the virus. And what the concern has been is whether this avian flu virus might in fact mutate in nature to affect humans, and so create another human flu virus and cause a pandemic. So there is interest in what happens in nature when viruses mutate. Two individuals were involved in grants from an agency in the United States, National Institutes of Health, and they were asked to answer the question whether the influenza virus can achieve sustainable aerosol transmission in ferrets. In other words, could this virus, which is normally in birds, eventually get to a point where an infected ferret could infect a non-infected ferret by aerosol transmission? In other words, could the virus mutate to some extent that it would affect ferrets? Now, why ferrets? Well, ferrets are animals like stoats. They have been used for this type of virology since the 1930s. They're a good experimental animal for looking at viruses that could affect humans. And the virus is whether it will affect the ferret, of course, is the issue. So what they did, and I'll talk briefly about both experiments in a moment. They did repeat passage, that's in other words, passed from one ferret to another. They were able to show that the replication of H5N1 in ferrets was an ideal model for studying influenza transmission in humans. Ferrets are much more likely than humans to have this disseminated form of the disease, multi-organ, and involving the brain as well. So it's very easy to assess ferrets and assess that they have become infected with the virus. And the work of these two groups was that they were able to show that mutant strains of the virus could be passed from one ferret to another. This was the experiment done by Koayoko. He's at the University of Wisconsin in the United States. And the other one was a person by the name of Foucher. and he is from Holland. They approached the problem in slightly different ways, but both approaches involved mutating the virus. In this particular procedure, they did random mutation to alter what's called the hemagglutinin, HA protein. The virus is composed of the outer shell, two components, hemagglutinin and neuraminidase. And depending on the type of hemagglutinin or neuraminidase, they will be coded accordingly. H5N1. The other one I will refer to is an influenza that affects humans. It's called H5N1. And what they did initially was they mutated the hemagglutinin component such that the mutated protein on the virus allowed it to attach to a receptor on the cell in the lung. So they looked at something that would attach to receptors in human upper airways, which is where you would be concerned that the virus might lodge and if it was going to cause a respiratory infection. What they then did was take this mutated gene, so the gene initially had two mutations. They took the mutated gene and they combined it with 7 genes from an H5N1 virus. And this is one that readily affects human lung. And the nature of genes and viruses, these are RNA viruses, they're not DNA viruses. The nature of the gene allocation in these viruses means that you can get recombination of genes quite easily. And the mixing of this mutated gene, the mixing of the mutated gene with genes from the H5N1 virus led to a third mutation. And this third mutation allowed the ferrets to become infected with the virus. And then those ferrets, in turn, the infected ones, were able to infect non-infected ferrets. And that process here, this fourth process, resulted in yet another mutation. So there were 4 mutations. And those 4 mutations changed the virus from being just something that generally affects birds to be something that would cause one ferret to infect another. And because that transmission could occur in ferrets, there's a real possibility that that infection would occur in humans as well. So the issue then became, should this information be made public? These 2 groups had done the research that was required of them by the National Institutes of Health. They had independently just found that mutations in the virus enabled it to infect a ferret and therefore possibly humans, and they wanted to publish. Like all scientists, they wanted to publish their information. The concern was that if you put this information out into the public domain, you're allowing people to see that these 4 mutations in a virus that hasn't yet, as far as we know, generally infected humans. You put this information about the mutated virus out there, so you tell people that how they can make a virus that could possibly cause a pandemic. We have no immunity to it, we have no resources for dealing with it. Should this information be made public? And I've already spoken about this 4th mutation. So, what I want you to do initially now is I've talked very briefly about what the virus does and how it has been mutated. What I want you to do as a group, I want you to teach me about virology. So I want group 1 to look up the information. I want you to use any electronic devices you have, and I want you to do a poster and group 1 to do virus attachment and cell entry, group 2 to do nuclear entry and transcription, group 3 to overlap a little bit and do transcription and replication, and group 4 to do assembly and budding. So you are back in the classroom again, your students. So I'd like you to use your computers or any other device. You can obviously take this information that appears on the internet and do a poster, and we'll put the posters up and you can teach the rest of us.